relatively short. But then we also, if you're still struggling to understand mitosis and meiosis because you're using the slides and there's not much about meiosis in the textbook, there's a couple links to some helpful videos on YouTube in the slides. And Paul recently put together an activity using playing cards to help you think about how that works. So we'll have that available for you as a tool to help you understand how meiosis works. If the lecture slides from yesterday and today and the homework for tonight aren't getting you there. I feel nervous about it.
And I made that up, so it might be useful, it might not be useful. I have no idea. If you do use it and you have feedback for how it could be more useful, then let us know. Those of you without masks, if you could find a mask somewhere. The reason I'm asking you to wear your masks is because in Missouri, there are still tons of people who are unvaccinated. Jewel is better at vaccination. Jewel faculty and staff are better than students at vaccination. We're at like 96% for faculty staff. Students are at 85%. As long as COVID continues circulating around, I'm concerned about your health and safety, which is the main reason. I'm vaccinated. I'm pretty sure Dr. Judd is as well. One of the frustrating things is that I don't know who in the class is vaccinated and who is not. And the student rate is about 85%, so you're about 10 percentage points behind faculty and staff. And I'm also not allowed by law to ask you what your vaccination status is. So all of you could be vaccinated, none of you could be vaccinated, 85% of you could be vaccinated. And so until we have this whole thing better under control, we would ask you to continue doing this ridiculous thing that is totally avoidable.
Normally we have a little set of backups here on the
Yeah, I'm
table.
not sure why we don't.
Today. I'll make sure we get them next time. Okay.
Trust me, I have a beard. I'm constantly getting beard hairs in my mouth. It tickles my nose and makes me sneeze a lot. Nobody wants to be free of these things more than me. And they fog up my glasses constantly, and they tickle my eyes, my eyelashes. Ugh.
Okay. Who can remind me at the beginning here the difference between meiosis and mitosis? Any teams that need to work on their points here? Yeah.
Meiosis is like sexual, whereas like mitosis isn't.
Okay, good. So mitosis is certainly involved in sexual reproduction, whereas mitosis is not. Can someone tell me about the difference between the end product of a mitotic versus a meiotic cell division? Yeah, sure.
Taking a shot in the dark here.
Yeah, good.
Is it that mitosis, when they separate, they form a completely like a clone of the same cell, and then meiosis, they like fold within each other, spread apart, and then it's a new set of.
End of the sentence? Yeah, sort of. So the first one, for mitosis, you're correct. You get two daughter cells that are clones of the original mother cell. Paige, you want to help them out on the end product of meiosis?
I was just going to add, like, mitosis is 2 diploid cells, meiosis is 4 haploid.
Haploid. Okay, someone explain what does haploid mean? Yeah. One set of chromosomes instead of 2, right? And chromosomes that come in 2, or chromosomes come in 2 because there are homologous pairs in each diploid cell. You get 2 versions of every chromosome in a cell. But if you make a haploid cell, you only get 1 version of that chromosome. That's because to complete a sexual life cycle, to make a new generation, you want the new generation to have 2 sets of every chromosome. So if you get 1 haploid cell from mom and 1 haploid cell from dad to make the cells that are your body, you're getting 1 set from mom, 1 set from dad, you end up with 2 complete sets. And you've recombined which genes make up you from two different individuals, from the parents, basically. So all this stuff about how mitosis and meiosis works, these diagrams we looked at last time here, all this stuff is relatively recent in our understanding, 1900s, mid-1900s, especially the 50s, when we really started, 30s to 50s, when we really started being able to see chromosomes under a microscope and understand what's happening as when these cells divide. But it was in the 1800s when Gregor Mendel was working with breeding plants and trying to work out how different traits that you might care about if you were breeding plants or breeding animals would be passed down. And we didn't really have a good understanding of why. It was hard to predict what offspring would look like, or if you're talking about cattle, the quality of their milk, things like that. It was difficult to predict just based on the qualities of the parents. And there were lots of people who were trying to come up with there was lots of interest, I should say, in trying to come up with some way of predicting what traits a next generation might have based on breeding. And it turns out that some of the math that he used came from probability theory that was developed for gambling. So people were wondering, what are the odds of mixing these things together and what do you get? The math that he used to work all this out came from probability theory and gambling. So here's a picture of Gregor Mendel. He was a Czech monk, I believe, and he was working with pea plants. It's pretty typical in biology to work with model systems. You all will learn about model systems later on, but basically it's sort of, say you want to know something about how some biological process works, well, let's pick an organism that we can work it out in, and then compare that one we understand in that organism to other organisms and see what we can learn from. So you might have heard of fruit flies, or C. elegans, this little nematode worm, or Arabidopsis, which is a little mustard-like plant. These are all classic model organisms that we use in biology today for genetics, development, even ecology and species interactions. But Gregor Mendel chose the pea as his model system. And here's a nice pink flower of a pea. Why did he choose peas? Because he could take the pollen from a single flower and use that pollen to fertilize the egg inside the same flower. So he could basically breed it with itself. And what that meant is within a generation or two, every parent looked identical to its offspring. Every offspring looked identical to its parents for generation after generation after generation. So he was able to basically select for a true breeding line that always produces seeds that will grow into a plant that he expects. It'll have pink flowers, it'll be this tall, it'll have this color seeds. All those features were the same every single generation, on and on and on, if he just kept selfing it. A lot of plants, like the goldenrod that you all have seen a bunch of out in the field, won't do this. They're self-incompatible. If you put the pollen of a goldenrod on its own stigma and then prevent any insects from visiting and bring another pollen, it will not set seed. But peas will do it. And then he selected a handful of traits that he wanted to be able to predict. So there's lots of attributes of a pea plant. He picked ones that would work for it. One of the features of these traits is they're categorical. A seed is either green or it's yellow. It's either wrinkled or it's round. There's some variation in height, but basically they were tall plants and short plants. And so these are the different traits that he was interested in predicting. And the location, or the locus, of the genes controlling for each of these traits were on separate chromosomes. Like, remember we talked about how we have 23 pairs? He picked genes that end up being on separate chromosomes. That turns out to be important, and we'll talk about why in a moment. So easy to measure, categorical, and on separate chromosomes. And you can use these true breeding. This allowed him to develop a model for predicting patterns of single gene inheritance. So one gene, two alleles, what's the pattern of inheritance? Well, the normal body cells of a pea plant, or in humans as well, are diploid, as we were just talking about. So each cell has two alleles of each gene. From every gene, those two copies that you have could be the same allele, or they could be two different alleles for the same gene. Each gamete produced by the pea plant, or by humans as well, contains only one allele for each gene, right? Because the gametes are haploid, so they're getting one of each chromosome pair. And then during fertilization, the chromosome pairs are reunited. In selfing, you're just putting the same pair that was in the parents back together in the offspring. So of course they're going to look identical every generation. So he worked out what the pattern is, and it wasn't until quite a bit later that another researcher named Reginald Punnett came up with a really useful graphical way of presenting that math. And so we're going to look at some Punnett squares as a way of predicting these things. But I want you guys to know how to use them well, be confident in your ability to use them. So I'm going to switch over to this board here. Let's say there's our little pea plant, right? So this pea plant is a diploid organism. But when it produces gametes, which are inside the pollen grains, those gametes are haploid. So let's say we've got a gene for flower color. What do we use? Pink and white? Yeah, pink and white. So I'm going to use, there's two notations people use for representing alleles. I'm going to show you one that's more general. So we're going to talk about a gene for flower color, and there's two versions of that gene. Color white or colored pink. So in this case, we have a plant that has two copies of the gene for flower color. And one of the things that Mendel noticed is that in each of these traits that he selected to investigate, when you had two different alleles, one of them was dominant over the other. So what that means, that tells you something about the relationship between the genotype and the phenotype. So a genotype refers to what combination of alleles do you have for a particular gene. So this is the genotype for this case. The color allele, you've got one of each of two possible alleles. The resulting phenotype, the outward expression of those genes, is the actual color of the flower. So I didn't bring my pink Sharpie today, my pink marker there using red. Pink. So what color is this supposed to be? Pink. Pink. Right. Oh, nice. Pink. Outward expression of the genotype. Now, what dominant means is that if you have a white and a pink, you go with the pink. Pink is the one that we see. Think of it as maybe this gene goes to our protein that successfully makes a pigment. In this case, a version of the gene is broken and is non-functional, and so it just doesn't make any color, and so it's not going to be pink. So if you have this allele, the flower can make the color, and the flower becomes pink. Other possible genotypes in this system, where there are only two possible alleles, would be having the same copy for pink color, two of the same, or having two copies that don't look the pink color that are white. So what's the alternative possible phenotype? White, right? And which of these two genotypes will give you that color? Well, right, yeah, number 2. So, 3 possible genotypes, 2 possible phenotypes, when you have a single gene, 2 versions, and one is dominant over the other. When it's time to make or to predict what kinds of gametes this plant will produce, we have to think back to meiosis. What happens in meiosis? What's the end product of meiosis? What kind of cells are produced by meiosis?
Haploid.
Haploid cells. And what do we call those haploid cells when you're thinking about sexual reproduction? Yeah. Gametes. So the first step in predicting the offspring of any given plant is to know what kinds of gametes it could produce for the trait. In this case, we're interested in flower color. So those are the 2 possible gametes. And you can even put a little circle around each of them to think, okay, I've made my gametes. Meiosis has occurred. Remember, in fact, there are 4 daughter cells in meiosis. But I don't have to write 4 here because 2 of them are the same. And in fact, the plant is making many, many thousands of gametes. So this is just representing the ratio. Half of the gametes are going to be for white color, half of the gametes are going to be for pink color. Now, there might be some other plant here that you're Gregor Mendel's student. And this one, I'm going to leave it as white. So what's the genotype going to be for this plant?
CW.
Yeah. So what are the possible gametes that that plant can make? Only see, only the color. What happens with fertilization? What does fertilization do with 2 gametes? It combines them, so the resulting cell from fertilization is diploid. So these are the resulting genotypes you can get with this process. Now, let's say this is the mother plant. She might produce 8 or 16 seeds. So this is just representing a ratio. 50% are going to be what color?
White.
50% are going to be
Pink.
So that's the process of going through and predicting these ratios. What you'll find is that you might cross 2 of these that are called heterozygous, meaning they have the 2 possible alleles, with another one also heterozygous. Then you have a 4x4 square. You might want to track 2 genes at the same time. Then you have 4 alleles to track, and you've got to predict gametes for those. So we'll do that up here. I will repeat this. Okay, so with these slides, if you download them, you should be able to step through this process just like I'm going to do here with you. So here's an example where we've got a trait of interest. What trait are we going to track here? What trait are we interested in?
Regular smooth.
Good, wrinkle smooth. I just want to make sure you're reading, right? Be in the habit of reading carefully the whole question, that kind of stuff. So what are the possible gametes that we can make out of a homozygous, round, or smooth seed like that one? The possible gametes.
R.
Just what case?
Uppercase.
One uppercase R. Good. And at the bottom?
One lowercase.
Ooh. Good. So think about what your possible gametes are. So how many different ways, how many different genotypes could you get by picking one from here and one from here? Here are a bunch of different. Now, one thing I'll point out is it doesn't matter what order they go in. So for example, over here, I wrote the rows first, and then I wrote the column here. But a CWCP is exactly the same. Just looking around things. So if you're picking one genotype, one allele from the top, and one allele from the bottom, Yeah, I'm seeing a few fingers up. This is the only possible solution of that. It's like having just one cell in that cross up there. But what Mendel did is he crossed these. He crossed, so this is the F1, the first filial generation of an experimental cross, where you're controlling who your parents and offspring are. So parents, F1, only one kind of possible solution, but then Mendel took these siblings and crossed them with themselves, crossed them with each other, to see what you would get if you cross siblings. Which sets up a very different square, a square with 4 possible solutions. So these are your possible alleles from one parent. These are the possible alleles from the father, the pollen. How many possible diploid genotypes can you make out of this cross? 3. Yeah, I saw lots of numbers up, and you all said it correctly. There are 3 possible ways of combining them. Homozygous dominant, homozygous recessive, or heterozygous. Now here's where I want to stop for a moment and remind you that over there I used the capital letter C for color as the gene of interest, and then I indicated with superscripts which allele I was talking about. In a lot of intro textbooks, you'll see it where they just pick a letter that's related to the phenotype, like R for round. They could have used W for wrinkled. And then they just use the case of the letter. But in systems where there's more than one allele in the population, it's useful to do it this way, and you all have actually probably are familiar with a system where we do that. Blood types. In blood types, there are at least 3 alleles floating around, and so we use this kind of notation instead of this. But all you have to do is ask yourself, how are we writing genotypes in this case? Are we using case, or are we using superscripts? Depends on the problem. And I just want you to be prepared to see superscripts someday in the future.
Superscripts are much more common in, like you'll see them much more common in genetics class, especially when you're working with things like model organisms like nematode worms and flies.
So what we can do now is compare the ratio of phenotypes that result from this cross to the ratio of genotypes that result from this cross. You all correctly noted that there were 3 possible genotypes. How many different phenotypes do you see for the texture? Yeah, 2. Good. I've seen a lot of fingers go up. Good. You're either round or you're wrinkled. So notice here we got a 3-to-1 for a 3-to-1 ratio for those 2 phenotypes. Whereas for the genotypes, we have a 1-to-2-to-1 ratio for those 3 genotypes that are possible. So get familiar or get comfortable with predicting both phenotype ratios and genotype ratios with this kind of a cross. So then the next step forward is you can start thinking about combinations of traits. You could be interested in, say you've got a plant that produces round seeds that are yellow, and another plant that produces wrinkled seeds that are green. In this case, we're using Y for the color gene and R for the texture gene. The challenge here is to think back to how meiosis works and how we make gametes if the genes are on different chromosomes. So what possible gametes would you get for this plant? How many kinds of gametes would you get for this plant? So each daughter cell, the end product of meiosis, is going to be haploid. See some different numbers out there. I want to let more people percolate. You're going to be haploid, and you're going to get 1 copy of each chromosome. Remember, the genes, in this case, are on different chromosomes. So, over here. our point, right? It's got a genotype. That genotype is R, 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 R. So that's one, that represents one chromosome pair, big R and big R. We're really representing one gene on that chromosome pair. This is the other chromosome pair. So if you imagine making a gamete from that, you gotta have one chromosome of each pair in the gamete. So how many R's do I put in there? How many Y's do I put in there? One. Could you get any other combinations of R's and Y's?
No.
So that's all there is to it. But what I want you to have when you, I mean, I could teach you guys to just, pick one from each pair, but I want you to be thinking about what's happening in the process of cell division, where you're shuffling those chromosomes and picking one of each pair. So, big R, big Y, down here is the same thing, but we're dealing with all recessives. You put them together, and you get an F1 generation that's heterozygous for both genes. It's got a big R and a little r, and a big Y and a little y. What if we cross these? How many possible gametes could you get out of this thing? How many possible gamete genotypes could you get out of that cell? Yeah, a good trick for this. How many of you heard of? Have you ever used the FOIL method in math class? We're going to do a different FOIL here. Pick the first allele of each pair. That's one gamete. Pick the outside allele from each pair. That's the second gamete. Pick the inside allele from each pair. That's the third gamete. And then pick the last, the little r, little y, from each pair. So I saw a bunch of 4s go up. 4 is correct. You get 4 possible gametes. There they are. Homozygous dominant, heterozygous with dominant for yellow, heterozygous with dominant for round, and homozygous recessive. And of course, Mendel's crossing these with themselves or with their siblings. So that's one set, there's the other set. How many possible solutions are there in this grid?
6.
How many possible cells are there in the grid? 16. Let's look at the ratios you get when you put them together, right? Notice when we put them together, we're keeping the genes grouped together. Sometimes I see people will kind of mix them up. They'll put RY, RY, and it can be confusing when you're trying to calculate your genotype and phenotype ratios later. So keep your genes together when you recombine them. It's like keeping the chromosome pairs together, the homologous pairs together. So what kind of ratios do you end up with? What's our ratio for yellow color to green color? Yeah. Yep. What's our ratio for wrinkled to round? Yeah. Yeah. 4, 16, do the math, reduce it, there you go. It's 4 out of 16. Is it 1/3? Yeah, wonderful. Yeah. But then we can look at the ratio of both traits together, and we find that if you cross a double heterozygote, a dihybrid cross here, with itself, you end up with a ratio of 9 to 3 to 3 to 1 for the combination, the combined phenotypes of interest. So there are 9 that are round and yellow. There are 3 that are yellow and wrinkled. There's 3 that are green and round. And there's 1 that's green and wrinkled. This is what you get when you do a dihybrid cross where you're tracking 2 genes and both parents are heterozygous. That's the predicted ratio. I could give you a different parent where one of these parents is homozygous for one of the genes, and you'll get a different outcome here, a different set of ratios. But it's pretty easy to just put them together as long as you keep your order straight, keep the order of your genes straight. So that's what'll be in the practice homework that I post on Moodle. Do a couple of these and see how they work out. When Mendel actually did this with real plants, he got 556 seeds in his F2 generation, that second set. And he got 315 that were round and yellow, 101 that were wrinkled in yellow, 108 that were round and green, and 32 that were wrinkled in green. And if you look at those ratios, we find that it's actually quite close to that 9 to 3 to 3 to 1 that was in the grid predicted by the Punnett square. And it's pretty simple to do some statistics that compare, is this 0.36 significantly different than just 3.0? And it's not, it's very close. And the larger the population, the larger number of offspring you get out of a cross like this, the closer that's going to approximate the ratio if you're dealing with a simple system. Single gene inheritance, the genes on separate chromosomes, and so forth. So the key for Mendelian inheritance is that each trait has a single gene point, and there's only 2 alleles in the whole system. And the probability of inheriting an individual allele from a parent is 50%. So when you make these gametes over here, pick one of these at random, 50-50 chance, and that's this. Pick one of these at random, 50-50 chance, that's this. So making the gametes is that 50-50 chance for each gene. But then putting them together in a new generation, you get these various other ratios to keep track of. We kind of already did that. Okay. So in comparing his results with the patterns he was seeing and the degree to which these were quite predictable, Mendel came up with 3 laws that he proposed to explain how phenotypes are inherited. And he said one of those is the law of the dominance. So for any pair of alleles, the expression of one trait conceals the presence of the alternate allele, which is what we were just doing with flower color at the beginning. That was the case for each of the traits he examined. The next was the law of independent assortment. So when you have 2 alleles, they're sorted into gametes independently of each other, and each gamete randomly gets one of each chromosome. He didn't put it in the phrase of chromosomes, he didn't know about it at the time, but we can do it that way to help you guys make the link to meiosis. And you can even figure out when that happens in meiosis, which phase. And then the last one is the law of segregation, which is just the diploid individuals pass only one of each chromosome on the next generation via their gametes. So this is just saying that basically the cells that you get at the end of meiosis are haploid. So when we talk about patterns of Mendelian inheritance, we're talking about systems that follow these rules, follow all 3 of these rules. Of course, Mendel didn't explain everything. There were lots of traits that didn't seem to fit what he was predicting. So we call all of that stuff patterns of non-Mendelian inheritance. And there's lots of ways that you could get non-Mendelian inheritance. One of them is, what if you've got 2 genes, but they happen to be on the same chromosome? So they're always going to be inherited together. They're not randomly assorting during metaphase because they're stuck together on the same chromosome nearby. So we call that linkage. That when 2 genes are close to each other in the genome, occurring on the same chromosome, that's a form of linkage. There's also incomplete dominance and codominance. I'll show you a picture of those. And then there are traits that are controlled by more than one gene. So Mendel was lucky that he had a height gene that was clearly like you're either tall or short. Most organisms, there's going to be a lot of genes that work together to influence height. Here's an example of codominance, where the phenotype includes expression of both alleles at the same time. So here we've got black chickens and white chickens. They're homozygous for feather color. But then if you cross them, you can get an erminette chicken that has both black feathers and white feathers. So you're just getting the outward expression of both alleles. Another example is incomplete dominance. So it could be that a gene that codes for a pigment gives you some amount of that pigment. The gene, having one copy of the gene means you can produce so much pigment through cellular processes. So if you've got 2 copies of the dominant allele, you're going to produce a lot of pigment. If you've got no copies of the dominant allele, you've got 2 recessives, you're homozygous recessive, you don't get any pigment. But if you're heterozygous, you get an intermediate amount of pigment. So this is digitalis, snapdragons. Yeah, snapdragons. And this is a common situation where you can get that. And now when you have codominance, you have this interesting situation where instead of having 3 genotypes and 2 phenotypes, you get 3 genotypes and 3 phenotypes, because you can tell the pink ones from the red ones. So notice how the ratios of phenotypes to each other are the same as the ratios of phenotypes to each other. All right. What about traits for which there are many genes that influence phenotypes? It was great if Mendel wanted to tell people how to breed all the green peas for their dinner that they want and never have any weird yellow ones mixed in there or wrinkled ones in there. But what if you're a farmer and you want to make sure that you've got the highest, fat content in your, or protein content in your milk, or some other trait that isn't so black and white, isn't categorical like that? Well, usually those kinds of traits that vary in a population are polygenic. They're traits that are influenced by many genes. And that's part of what gives you this continuous variation for a lot of genes that have quantitative variation to measure the degree to which there's protein in the milk, or the degree to which the flowers are red, if there are many genes influencing red color. And you could have lots of different pairs of dominant and recessive alleles for each of these genes. So here I think is where our slides overlap, Paul.
Yeah.
I think I should turn it over to you. I want you guys to
Do you
be
have
prepared to.
Do you have the remainder of the?
This one?
Oh yeah, I'm gonna
Okay.
get out and I'll get back in.
So while we switch computers here, I hope that walking through this carefully, remember your first step is to think about what your possible gametes are. Circling them helps you remember that you've made your gametes. Put them across the top, put them across the bottom, and then draw your square. And then combine it together, keeping your homologous pairs together instead of mixing them up.
That was the fastest you've ever gone through that.
Yeah, it was. But I think
Was
we're
it
doing
too
all
fast?
No.
Okay.
I'm really trying to make it more efficient on the board because I knew that we wanted to get through this. Okay. I think better than last time.
All right.
And they got the homework.
And I have their exams.
The homework, turn it in next time so I can give you guys feedback and make sure you're getting the fun square stuff.
Did you do all of those?
What's that? I
Did you
haven't
do all
graded
of
any of
them?
them. Oh, yeah.
Okay, so Dr. Judd took you through a dihybrid cross, and you get this symmetry to the Punnett square that he was talking about, where you have the homozygous dominants in the upper left-hand side of the table, you have your homozygous recessives in the lower right-hand side of the table, and as you move diagonally across that table, you get fewer and fewer dominant alleles. And he was looking at traits where you have these discrete sorts of characters. You're either yellow pods or green pods, yellow seeds, green seeds, round seeds, wrinkled seeds, etc. You'll remember a couple of weeks ago, I had you guys all get up and stand, and we put the tall people at one end of the room, we put the short people at the other end of the room, and you saw that there is this kind of continuous variation in human height. We have short people, we have tall people, we have all kinds of heights in between. Well, your height is underlain by genes in the same way that round-seeded versus yellow seeds are underlain by genes. And you have different varieties of those genes, different alleles of those genes that control your height. But in this case, those genes are governed by lots of different genes, all of which contribute a little bit to the phenotype. And so it's like adding weights to a scale. You put one dominant allele on there, it makes it a little heavier. Another dominant allele makes it a little heavier still, until you've got all of the dominant alleles, and that's where you're at your heaviest. Same thing for height. Individuals with 0 dominant alleles are very short. One dominant allele makes you a little taller. A second dominant allele makes you a little taller. So the actual identity of the gene isn't as important as much as it is how many dominant alleles you have. So the next thing that Dr. Judd was going to be talking to you about is this idea of polygenic inheritance, traits that are underlain by the action of multiple genes acting on that single trait. So my first goal is to get across to you how this all works, especially in terms of the inheritance of polygenic traits. In the process of doing this, we're going to talk about Hardy-Weinberg equilibrium because it's useful for thinking about evolution, and we're going to use that in the context of selection because ultimately we want you to understand how selection is acting on these birds. Now, beak size, you didn't just have birds with big beaks and little beaks, did you? You have all this continuous variation. So if there is heritability, if beak size and shape are heritable traits, more than likely it's going to be one of these traits where a bunch of different genes all control size and shape of the beak. And so it's going to be a polygenic inheritance sort of situation. I'm going to introduce you to this thing called the breeder's equation, which allows us to project into the future what happens with evolutionary change in a trait, and then show you how we can analyze some of these things, like know what the strength of heritability is, for example. How heritable is a particular trait refers to how similar the offspring are going to be to their parents with respect to that trait. So Dr. Judd was about to get to this point. So we had a monohybrid cross that dealt with only one trait. We had a dihybrid cross that dealt with 2 traits. This is now a trihybrid cross because we're now dealing with 3 genes, gene A, gene B, and gene C. So in this case, we have a homozygous dominant individual for all 3 genes, and we have a homozygous recessive for all 3 genes, and we are going to cross those individuals. Now, in this case, this is now a polygenic trait, not a discrete trait like Dr. Judd was talking about. So imagine that this trait is height. And if you are homozygous dominant for all 3 genes, you are the tallest individual in the population, and we're going to cross that individual with the shortest individual. And to even make this more specific, let's actually put phenotypic values on these individuals. So this individual is 1 unit tall, whatever the unit is. Could be an inch, could be a foot, could be whatever. This individual is 7 units tall because a homozygous recessive is 1 unit tall, and each of these dominant alleles adds 1 unit to your height. So there's 6 dominant alleles, so you're 7 units tall. What's the first step when you do a genetic cross? What's the first step after you know what the parents are? Get the gametes. What would the gametes be in this case for this individual? Yep, a capital A, capital B, capital C. I don't know, I wasn't listening as closely as I could have. Do you usually circle your gametes?
Yeah, this time I told him make sure to circle it this time.
Yeah, I always circle my gametes because then it reminds me that that's a gamete, not an individual. And so, this is a gamete. Is it diploid or is it haploid? It's haploid. It has half the complement of chromosomes that it had before. What's the gamete composition for this individual? And when we combine those through fertilization, what are we going to get for the genotype? Heterozygous for A, heterozygous for B, and heterozygous for C. That's a new individual. How tall is this individual going to be?
4
4 units tall, which is actually the average size of averaging those two parents. Back when Darwin was thinking about inheritance, one of the stumbling blocks he had was this idea of blending inheritance, that when two individuals get together and mate, they produce offspring that are a blending of their phenotypes. And what he didn't recognize at the time, because they hadn't discovered genetics yet, was that this blending kind of inheritance, where you take the tall individual, you take the small individual, and you get this intermediate height individual out of that coupling, that stuff that looks like blending inheritance is actually just the inheritance of polygenic traits. But because we didn't even know what a chromosome was, we didn't know what genes were, it hadn't even been discovered yet. It wasn't discovered until 1866. He published The Origin of Species in 1858-59. There was no way that he could have thought about blending inheritance in a rigorous, modern sort of way. So now we have a whole population of offspring that are intermediate height individuals. They're exactly the average, and every one of them is exactly the same height. Now we can take these individuals and we can allow them to interbreed with one another. So we'll take heterozygous individual for both traits, cross it with a heterozygous individual for both traits. What's the first step when we're doing a cross? Make the gametes. How are we going to make the gametes now in this case? We have more to work with. What's that? Okay, but before we make the Punnett square, we need to know how big the Punnett square needs to be. So the first thing we can do is foil. How do you foil this thing? My advice would be to just foil the first part. Put the dominant allele for the third gene on it, and then just foil it again so that you can then get the recessive allele. So you have 8 different gamete types, and that's now all the possible combinations. It's just a modification of the FOIL method that Dr. Judd told you. So we have 8 possible gametes from one parent, and we're going to have 8 possible gametes from the other parent. So this is what we've done. And this is the Punnett square. So what would we do to fill in this Punnett square?
Just match
Yeah, I do it a little more systematically.
each one.
What I typically do is I'll take these gametes and bring them across. These gametes, bring them across. These gametes, bring them across. And then I take these gametes and bring them down. So take the next 15 or 20 minutes and work through that Punnett square in your notes. That's amazing. You guys think I'm serious about that. No, don't do that. That takes forever.
I was trying to get going.
This is what it would look like if you brought the gametes across and then brought the gametes down. It's big and complicated, isn't it? But what do you notice about it? What's that? Other than it's big and complicated. Hmm? There's repetition. So we have individuals that have the same genotype right here and right here. What else do you notice about it? You also have the heterozygote all the way across the middle diagonal. Just like we did when we did a monohybrid cross, just like we did when we did a dihybrid cross. What else do you notice about it? Lots of combinations, but not 64 combinations. That's what the repetition, the redundancy is about. What's in the upper left-hand corner? Homozygous dominant for all 3 genes. What's in the lower right-hand corner? Homozygous recessive for all 3. The diagonal is heterozygous for all 3. But once again, what matters is not whether you have a dominant A allele or a dominant B allele or a dominant C allele. What matters is how many dominant alleles do you have in total. So going back to this symmetry of the table that I was talking about, The first row is 6, 5, 5, 4, 5, 4, 4, 3, dominant alleles. The first column, 6, 5, 5, 4, 5, 4, 4, 3. The second column, 5, 4, 4, 3, 4, 3, 3, 2. The second row, 5, 4, 4, 3, 4, 3, 3, 2. You go from a lot of dominant alleles in the upper left-hand corner to no dominant alleles in the lower right-hand corner. And as you move diagonally across this table from upper left to lower right, you get fewer and fewer alleles. Now, how many homozygous dominants do we have for all three traits? How many individuals do we have that have 6 alleles who are 7 units tall? Only 1. How many homozygous recessives do we have? Just 1. How many individuals do we have with 1 dominant allele? 1, 2, 3, 4, 5, 6. How many with 2 dominant alleles? This gets more complicated, so I'm actually going to do it on the board. Oh. 2 dominant alleles. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 0 dominant alleles was 1 individual. 1 dominant allele was 1, 2, 3, 4, 5, 6 dominant alleles. 2 dominant alleles is 15. 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 dominant alleles. How many individuals have 3 dominant alleles? One, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Remember, that's the average phenotype. 1, 2, 3, 4, 5. That's now 20 dominant alleles. What's the other half of this graph going to look like? It's going to look exactly like that half. Why? Because this thing is symmetrical. So we have 5 dominant alleles, 6 dominant alleles. This will be 15. This will be 6. Whoa, what happened?
You missed 2.
Oh, I missed 2. I missed a 2. 2, 3, 4. 3 dominant alleles, which is a height of 4, 5. And 6 dominant alleles, which will be 1 individual. What does this start to look like?
Unimodal.
It's a unimodal curve, but it's a specific type of unimodal curve. It's a normal curve. It's that curve of those students standing on the football field that Dr. Judge showed you a couple of weeks ago. It's that normal curve. And if we were to plot your height in a frequency histogram like this, it would look like a normal curve. If we were going to plot beak morphology, beak size, it would be a normal curve, unless it's a bimodal curve because natural selection is acting on it. This individual is 1 unit tall. These 6 individuals are 2 units tall. These 15 individuals are 3 units tall, 4 units tall, 5 units tall, 6 units tall, 7 units tall. The number of dominant alleles you have adds something to your phenotype. That's why we call these traits, not only, we call them quantitative traits, but I think a more descriptive term for them is they are additive genetic traits. Because that gets across this idea that each dominant allele adds a little to the phenotype. These are the kinds of traits, these traits that are underlaid by multiple genes where each dominant allele adds a little bit to the trait. These are the types of traits that make up really ecologically relevant sorts of traits, like height, weight, sprint speed, beak size, beak shape, etc., etc. Peas have green pods. Well, really and truly, who cares? Does that really have any effect on their evolution? They have green seeds. If an animal is going to eat the seeds because they're in a green pod, that might have a value. but the seeds inside, there's not really any selective value that I can think of in terms of the seeds themselves being green. And you're the plant biology guy, what's your thought on that?
It affects how long the seeds live, but it's gonna be minimal.
Yeah. So we, as ecologists and evolutionary biologists, we're interested in the types of traits that are important for survival and continued reproduction and continuing the persistence of a species in a landscape. And oftentimes the types of traits that are important for that aren't these discrete kinds of traits. They actually end up being these additive genetic types of traits. One of the things that I want you guys to get across, to remember, to take home from this lesson, is that when you see a trait distribution in a group of organisms that looks like this, think, Mendelian genetics, Mendelian style inheritance for a trait that has a lot of genes contributing to that trait. That's what led to this type of a distribution. Later on, we're going to see how this distribution changes shape under the influence of selection. But we'll put that off until Friday. We'll talk about that.
Now that you understand Mendelian genetics and how it helps us understand distribution of traits, you can see why without Mendelian genetics, it was so hard to figure out how these quantitative traits are interacting in the wild or in farmers or in plant breeders back in the time of Mendel and before, when they were trying to
I teach
predict things.
I teach a CTI class called the Ecology of Food, and we're now going through the genetics portion of things. And when I talk to them, I talk to them about like how much meat you can pack onto a beef cattle. That's a quantitative genetic trait. I talk to them about how much milk a milk cow will produce. That's a quantitative trait. How many kernels of corn are on a cob of corn? That's a quantitative trait or an additive genetic trait. There are tons of these traits out there, and many of these traits are really important to farmers in many cases. Don't go left, don't go yet. So these quantitative traits are determined by multiple genes, each dominant allele adding to the additive effect of the genes on the phenotype. We actually use this, we analyze these kinds of traits using a subdiscipline within genetics called quantitative genetics. It's a highly statistical field because we're not really all that concerned about what the underlying genes actually are. We're concerned about how these distributions of phenotype behave as they're being inherited and being selected on. And so we actually use a lot of math in doing this. And we can think about this in the context of a simple cross. So when you think about just a simple cross with, let's just say we're looking at the A gene, how would we make gametes from this? Just be a big A and a little a. When those are fertilized, you get a heterozygote. What's the next step in a typical kind of textbook sort of cross?
Breed that one.
You allow those to interbreed, those offspring to interbreed. You get this cross, which leads to this Punnett square, which is a Punnett square that you've already seen from Dr. Judd. But this is only one way of thinking about a Punnett square. You can also think about the Punnett square from the standpoint of how many alleles in the population are represented by the dominant allele versus the recessive allele. So up here at the beginning, we had a population of 2 individuals. How many alleles do we have total in this population?
2.
We have 4 total. How many of those are the dominant allele?
2.
2 out of 4, so 50% of them are the dominant allele, 50% of them are the recessive allele. When we go through and let those individuals mate, now we're allowing a population of individuals that all have the same genotype. How many alleles do we have here? 4 alleles. How many of them are the dominant allele? 2. How many of them are the recessive allele?
2.
Same thing, 50-50 mix of allele types. We allow them to breed, we get a new generation of offspring. How many alleles do we have in this new population, this new generation? We have 8, 2, 4, 6, 8. How many of them are the big A allele? 4. How many of them are the little a allele?
4.
So we can basically figure out what the proportion of these genotypes are going to be by replacing the actual alleles with their frequency. So in this case, P is the frequency of the big A allele, Q is the frequency of the little a allele. This would be mean that there's a, well, so to figure out what's in this square, you would take the frequency of the one multiplied times the frequency of the other, that's P squared. Frequency of the little a allele multiplied times the frequency of the big A allele would be PQ. PQ would be here and Q squared would be here. And that tells you what the frequency of the homozygous dominant is, what the frequency of the heterozygote is, and what the frequency of the homozygous recessive is. And it's 2PQ because in this Punnett square, you have a PQ here and you have a PQ there. So now we can think about allele frequencies and whether or not allele frequencies change over time, because we now have a rough mathematical way of tracking this. We'll start here on Friday. No homework from me to get ready for Friday.
Just the practice Punnett squares.
Just bring your brains. We have your exams.
Oh, thank you.
I have this half. You want to get the other half? I have it right there. Take it. Jamie.
We gotta take these home again.
Why? What?
Do we take them home again?
Yeah. No, I don't want them. I didn't want to grade them in the
Some
first
teachers.
place. Hey,
Just collect them.
Mia. Yeah. Think I'm that much of a glutton for punishment?
I don't know.
Trevor. David.
Or some part of.
Oh, yeah, you're over here. Savannah. Maggie. Where did Emma go?
She left.
Yeah, she didn't tell me she was going to be late.